Hello, my name is Tomasz and I work in the field of AAV-based therapies, specifically on the downstream process team. Today I want to share with you how advanced analytical methods are transforming our daily work and helping us achieve remarkable results. Therapeutic applications of AAV rely on the removal of process and product-related impurities, which pose serious safety threats and burden the economics of manufacturing. The most critical process-related impurities include host cell impurities such as host cell DNA, host cell proteins, and chromatin complexes, as well as plasmids and transfection reagents. Critical product-related impurities include non-potent AAV capsids such as empty AAV, partially filled AAV, and AAV capsids containing host cell DNA inserts, as well as capsid-to-capsid complexes or aggregates, and capsid DNA complexes. In this product walkthrough, we will explore how PATfix, with its multi-detector setup, can monitor the removal of these impurities throughout the entire AAV downstream process. We know that the fastest way to develop and optimize a process is by utilizing extensive analytical insights leading to effective and safe therapeutic agents. PATfix allows me to gain important insights while optimizing process steps, helping me to complete crucial client projects to the desired specifications and timelines. In-process analytical methods differ from QC analytical methods as they need to focus more on speed and throughput, generating the data required to support immediate process decisions that often need to be made on the spot. One of the biggest challenges we faced was the ability to analyze all DSP samples, including crude samples, without having to extensively prepare them for analysis. Having the ability to analyze pre-capture samples has two key advantages that ease subsequent DSP steps. The downstream process is always optimized to a specific starting material with target AAV full capsid titer, volume, and both process and product-related impurity profiles. By analyzing the starting material, such as cell lysate with UV fluorescence and multi-angle light scattering detectors simultaneously, it becomes much easier to adjust the downstream process steps to the quality of the starting material. Likewise, requirement ranges can be put in place to ensure only material with sufficient quality proceeds further downstream. Then there is the optimization of pre-capture process steps like filtration and TFF. A lot of losses and gains can happen during these two important early DSP steps. Using PATfix and the triple detectors, I can easily compare recoveries and impurity removals between different consumables and process parameters to find the optimal strategy. The light scattering detector is particularly important for crude samples as it allows for specific tracking of particles like AAV capsids. At the same time, UV and fluorescence detectors provide valuable information on the removal of all other protein and nucleic acid impurities. Tryptophan for proteins and picogreen for DNA detection at the same time allows me to quickly see any residual impurities that might be present. This is enabled by the fluorescence detector that can monitor two simultaneous channels. UV 260 and 280 nanometers wavelengths detect all protein and nucleic acid components in a sample. This combination of detectors, together with the chromatographic separation, makes it very easy to identify and track all process and product-related impurities. This is especially useful since the full AAV capsids, our product, are always visible even with the impurities present. In post-capture samples using the triple detector on the PATfix system, we can precisely determine not only full and empty AAV capsids, but also other subpopulations that are becoming increasingly important to remove. Compared to other analytical methods, preparing samples, even crude samples, is very easy. It usually involves only dilution with loading buffer, which saves a significant amount of time and effort. Using the included AAV standard and method, I can always check that the system is performing to expectations. This makes it much easier to trust and act on the results obtained. Additionally, I can prepare a calibration curve using the AAV standard, allowing me to determine capsid titer in post-capture samples. One of the features I appreciate the most is having one software for running all the hardware, including all of the detectors, as well as analyzing all the detector data obtained for the samples. This makes it easy to analyze results, share them with colleagues, and present findings to higher ups during meetings. Other solutions often require multiple software programs to obtain the information I need. The system and software are certified according to FDA and EMA regulations, which is important for a few key reasons. Access to data is easy to restrict only to the people in the project group, and all the data obtained is safely stored with access and tamper protections. This is a necessary feature in our industry. The software also allows me to connect from the office, which is great for checking in on progress remotely and analyzing results without having to be in the lab. This flexibility is invaluable for maintaining productivity and ensuring timely decision making. Since I'm using the same package of system, column, buffers, and method that has been validated by my colleagues, I don't have to worry about my analytical results being skewed. This helps me focus on my important work of optimizing the purification process for AAV-based therapeutics. PATfix enables real-time monitoring of AAV samples, allowing me to track changes in impurity levels and the ratio of empty to full AAV capsids throughout the purification process. This real-time data helps me identify potential issues early so that I can make timely adjustments to the process. The ability to monitor these parameters with minimal sample handling required is not only a quality of life improvement, but also helps prevent errors. In turn, it is much easier to streamline the purification process and reduce the time and cost of producing high-quality AAV therapeutics. Rapid chromatographic analysis of AAV fractions removes a bottleneck in fast process development. Sartorius Bioseparations has developed the PATfix system specifically for the analysis of large biomolecules and in-process control of upstream and downstream process steps. With its validated methods together with orthogonal analytical approaches, the PATfix system offers a comprehensive solution for insight into the AAV process. It enables fast and precise process monitoring, which is crucial for the successful optimization of gene therapy vector production processes. To learn more about the PATfix AAV platform and its capabilities, visit our website, try out the software demo, or contact our team for more information.